Sit up straight right now. Not because your mother told you to, because what happens to the disc between your third and fourth lumbar vertebra in the next 60 seconds of how you're sitting is the difference between a spine that functions at 35 and one that functions at 55. You probably just moved. Good. Hold that position because by the time this is over, you will never sit the same way again, not because someone told you posture matters, but because you will understand exactly what is happening inside the disc while you slouch. The fluid leaving it, the cells dying inside it. The reason the damage does not hurt until it is already a decade old. The spine does not warn you. That is the problem. It collects damage silently in tissue that has almost no nerve supply for years before the first symptom arrives. And the mechanism driving that damage is not age. It is not genetics. It is not how much you lift or how hard you train. It is how long you sit still. Your spine contains 23 intervertebral discs. Each one is a hydraulic structure, not a cushion, not a pad, not a shock absorber in the passive sense. A living hydraulic system with its own internal pressure, its own fluid dynamics, and its own biology that depends entirely on movement to survive. The disc has two parts. The nucleus pulposus at the center, a gel-like core made of approximately 80% water, and a mesh of proteoglycan molecules that attract and bind that water with extraordinary force. And the annulus fibrosus surrounding it, concentric rings of collagen fibers arranged at alternating angles like radial tire plies, designed to contain the pressurized nucleus under load. The nucleus is under pressure right now. Sitting upright, the intradiscal pressure in your lumbar spine is approximately 150 kilopascals. Standing, it drops to around 100. Lying flat, it falls to 25. Slouching forward, the position most people default to within 11 minutes of sitting, it climbs to over 200 kilopascals. That number matters. Not because pressure damages the disc directly, but because of what pressure does to the only nutrition system the disc possesses. This is the fact that changes everything. Every other tissue in your body is fed by blood vessels. Capillaries reach within micrometers of every living cell in your muscle, your liver, your skin. Oxygen arrives. Glucose arrives. Waste products leave. The system is continuous, automatic, and does not require your cooperation. The intervertebral disc is the largest avascular structure in the human body. No blood vessels penetrate it. Not one. After approximately age 8, the tiny capillary buds that fed the juvenile disc are gradually occluded and resorbed. The adult disc sits inside your spine, completely cut off from the circulatory system that feeds every other tissue you have. So how does it eat? The cells inside the disc, the chondrocyte-like nucleus cells, and the fibroblastic annular cells that maintain the collagen architecture survive through diffusion. Nutrients from the blood supply and the vertebral end plates, the thin cartilaginous caps above and below each disc, diffuse down a concentration gradient through the disc matrix to reach the cells at the center. The distance from the end plate to the center of a lumbar disc is approximately 8 millimeters. 8 millimeters through an avascular matrix fed entirely by diffusion. The cells at the disc's center are the most nutritionally remote cells in your body. They live at the absolute margin of what diffusion can sustain, and diffusion is not passive in this context. It requires something to drive it. That something is movement. When you move, when you walk, when you shift weight, when you go from sitting to standing, the disc compresses and decompresses rhythmically. Under compression, fluid and metabolic waste products are squeezed out of the nucleus through the end plate. Under decompression, the proteoglycans in the nucleus, those water-hungry molecules, pull fresh fluid back in, dragging nutrients with it. The disc breathes through movement. Compression, decompression, squeeze, rehydrate, load, unload. Each cycle exchanges the disc's internal fluid. Each cycle delivers glucose and oxygen to the cells at the center. Each cycle removes the lactic acid and carbon dioxide that those cells produce. This is the pump, and it has no muscle. It cannot run itself. It runs only when you move. When you sit still for 90 minutes, the average uninterrupted sitting duration for a desk worker, the pump stops. Not slows. Stops. The nucleus cells continue consuming the oxygen and glucose that the last movement cycle delivered. The lactic acid they produce accumulates. The pH inside the disc drops, and the cells, sensing the acidic environment, begin reducing their metabolic activity, producing less collagen, maintaining less proteoglycan, pulling less water, the nucleus begins to dehydrate. You cannot feel this. The disc interior has no nociceptive nerve fibers. There is no pain signal, no warning, no indicator that the hydraulic structure between your vertebrae is sitting in its own metabolic waste, slowly dehydrating while you finish the meeting, the episode, the scroll. Now add the position. A slouched lumbar spine, pelvis tilted posteriorly, lumbar lordosis flattened or reversed, does not distribute load across the disc evenly. The anterior portion of the disc, the front face, carries the majority of compressive load. The posterior annulus, the back wall of the disc, is placed under tensile stress. At 200 plus kilopascals of intradiscal pressure in a slouched position, the posterior annular fibers are being stretched while the anterior nucleus is being compressed. The annular rings, those alternating collagen plies, experience differential loading they were not designed to sustain continuously. Collagen under sustained asymmetric load undergoes a process called creep. The fibers don't snap. They yield slowly, microscopically, rearranging their internal structure under the sustained mechanical input. Tiny radial fissures form in the annular layers. Not tears, not herniation. Fissures, microscopic failures in the collagen architecture that accumulate over months and years of repeated asymmetric loading. The disc is not exploding. It is losing structural integrity one microscopic layer at a time. And here is what makes this insidious. The annulus fibrosus has nerve supply only in its outermost layers. The inner two-thirds are as innervate as the nucleus. Those fissures are forming in silence, in tissue that cannot report pain, in a structure that will not announce its degradation until the fissures reach the outer layers or until the nucleus, dehydrated and increasingly fibrous, begins to bulge against the neural structures adjacent to it. By the time your back hurts, the disc has typically been accumulating damage for 7 to 10 years. The spine aged in silence. For a decade, Wilkie and colleagues, publishing in Spine, measured intradiscal pressure across a full range of positions and activities. The data rewrote the clinical understanding of spinal loading. Relaxed standing produces less disc pressure than sitting. Walking produces less than sitting still. And the position that produces the highest lumbar disc pressure, higher than lifting a 20-kilogram weight with proper form, is sitting slouched while leaning slightly forward. The position most people assume within minutes of sitting down. Sato and colleagues demonstrated that sustained static loading of the disc reduces its water content measurably within 20 minutes. The hydraulic height of the disc, the distance between the vertebrae it separates, decreases. You are measurably shorter after sitting for 2 hours than you were before you sat down. The disc has lost fluid. You regain it overnight when horizontal loading removes the compression and the proteoglycans rehydrate. But the rehydration requires a disc with intact proteoglycan architecture to drive it. As the disc ages, as the proteoglycan content decreases, as the nucleus becomes progressively more fibrous, the overnight rehydration becomes less complete. The disc that starts each day slightly thinner than the day before. Urban and Holm demonstrated that the oxygen concentration at the center of a human lumbar disc during sustained static loading falls below the threshold that nucleus cells require for aerobic metabolism. They shift to anaerobic metabolism. Lactic acid production accelerates, pH drops further. Below a critical pH threshold, the enzymes that maintain disc matrix, the ones building and repairing the collagen and proteoglycan architecture, lose their function. The disc is not just dehydrating. Its repair mechanisms are shutting down. Sustained sitting is not inactivity. It is an active biological process of accelerated disc degradation driven by the cessation of the only nutrition system the disc possesses. The research converges on a number, 30 minutes, not the number of minutes you should exercise, not the number of minutes of walking required, the number of minutes of uninterrupted sitting after which disc nutrition begins measurably declining. The point at which the diffusion gradient that feeds the nucleus cells starts reversing, waste products accumulating faster than the residual fluid exchange can clear them. At 30 minutes, the pump needs to run again. Not for an hour, not for a structured exercise session, for 90 seconds. The disc needs one movement cycle sufficient to recompress and decompress the nucleus, to exchange the accumulated fluid, to reset the nutritional gradient. Stand up, sit back down, walk to the kitchen, take the stairs once. Not because of cardiovascular benefit, not because of caloric expenditure, Because the avascular disc between your lumbar vertebrae has been running on the nutrient reserve from the last time you moved, and that reserve lasts approximately 30 minutes before the biology inside the disc begins shifting toward degradation. The intervention is not a workout, it is a biological maintenance cycle for tissue that cannot maintain itself. The disc is not the only structure aging in silence. The facet joints, the paired articulations at the back of each vertebral level that guide spinal movement and share compressive load are also responding to what sitting does to spinal alignment. In a posteriorly tilted pelvis, the lumbar facet joints are loaded asymmetrically. The cartilage surfaces that line them, identical in composition to the cartilage in your knee, experience shear forces that upright spinal alignment distributes more evenly. Sustained asymmetric loading of cartilage produces the same creep phenomenon as the annular fibers. The cartilage matrix slowly yields under forces it was not designed to sustain continuously. The deep spinal stabilizer muscles, multifidus, running from vertebra to vertebra in short segments, providing the segmental stiffness that protects the disc and facet complex, experience something different. They do not degrade under load, they degrade under the absence of it. In a slouch spine, multifidus is neurologically inhibited. The muscle is switched off, not fatigued, not damaged, not weak in the conventional sense. Inhibited. The proprioceptive feedback from the overstretched posterior ligaments signals the nervous system to reduce multifidus activation. The muscle that is supposed to protect the disc is shut down by the same position that is degrading it. The disc is dehydrating. The facet cartilage is creeping. The primary stabilizer that should be guarding both is neurologically off, all three simultaneously, all three silently. In the position you were sitting in when you started reading this, disc degeneration is graded on the Furman scale, a 5-level MRI classification from normal hydrated nucleus to complete disc collapse. Population studies show that grade 3 changes, moderate signal loss, disc height reduction, early structural compromise, appear in the lumbar discs of sedentary adults at a median age of 43. In age-matched populations with high daily movement variability, grade 3 changes appear closer to 53, a decade. On imaging, visible on MRI, in the same vertebral levels, in people the same age. The difference is not genetics, not occupation, not trauma history. It is the daily movement pattern that either runs the disc as nutrition pump or leaves it stopped. Your disc does not know your age. It knows whether the pump ran today. The intradiscal pressure data is direct measurement. Wilkie's laboratory used pressure transducers implanted in living discs. The nutrition diffusion data is established biochemistry. Urban's work on disc oxygen gradients is foundational to the field. The creep data in the annular fibers is mechanical measurement on cadaveric and living tissue. The 30-minute threshold is a synthesis of multiple studies rather than a single landmark measurement. The specific number has not been confirmed with the precision of the pressure data. The direction of the evidence that uninterrupted sitting of 30 or more minutes produces measurable nutritional decline in the disc is consistent across studies. The exact threshold may vary by individual anatomy, disc health, and spinal loading pattern. The facet and multifidus inhibition mechanisms are established in the musculoskeletal literature. The simultaneous triple failure, disc facet stabilizer, during sustained slouched sitting is a synthesis of 3 separate lines of evidence rather than a single study demonstrating all 3 concurrently. The viewer should know which claims are direct measurement and which are convergence. The disc pressure data is direct measurement. The 30-minute synthesis is convergence. Both justify the same behavioral conclusion, but they sit on different shelves. The spine that functions at 55 instead of 45 does not belong to someone who exercises more than you do. It belongs to someone who interrupted their sitting before the pump stopped working. Not dramatically, not with a standing desk or an ergonomic overhaul or a structured stretching program. With 90 seconds of movement every 30 minutes, a biological maintenance interval for the only major structural tissue in your body with no circulatory backup system. The pharmaceutical industry has no product for disc degeneration. There is no drug that restores proteoglycan content. No injection that rehydrates the nucleus. No procedure that reverses the collagen fissuring in the annular layers. When the disc is gone, the options are management and replacement. The biology does not run backward. The maintenance window is now, while the disc still has the architecture to rehydrate, while the proteoglycans still have the capacity to pull fluid back in, while the pump, stopped for the last however many minutes you have been sitting, still has something to pump. Stand up. Not because your posture is bad, because 8 millimeters of avascular matrix is waiting for the one input it cannot generate for itself. You are the pump. The disc does not age on a calendar. It ages on a movement schedule. Run it.